Why do we age? Why does our skin wrinkle, our hair turns gray, and our bodies slowly weaken with time? For centuries, humans have searched for the secret of eternal youth. Kings, philosophers, and scientists alike have wondered whether aging is inevitable, or if one day we might overcome it. Today, we know that aging isn't just a matter of years passing. It's a biological process written into the deepest layers of our cells. It begins silently, often in midlife, long before we notice the first wrinkle or gray hair. And at the heart of it all are our genes, the blueprint of life, shaped and influenced by the environment we live in. In this video, we'll explore how our DNA, our epigenome, and our lifestyle interact to drive the aging process. We'll look at the genetic players that control longevity, how DNA damage accumulates, and how science is trying to slow, or even reverse, the process of growing old. Aging is the gradual decline of our cells, tissues, and organs. When we're young, our bodies function with extraordinary precision. Cells divide efficiently, wounds heal quickly, and our systems protect us from disease. But as time passes, our cells begin to struggle. They don't divide as often, they make more mistakes, and the systems that once protected us begin to break down. Aging isn't just about looking older, it's about the slow decline of our biology. And the instructions for all of this, both the growth and the decline, are written in our genes. Now let's dive into some of the most important genes that influence how we age. One of the most fascinating players is telomeres, the protective caps at the ends of our chromosomes, like the plastic tips on shoelaces. Each time a cell divides, its telomeres get a little shorter. When they become too short, the cell can no longer divide safely. Scientists often call telomeres our biological clock, slowly ticking down as the years pass. Helping maintain these telomeres is an enzyme called telomerase. Telomerase rebuilds the ends of chromosomes, essentially resetting the clock for cells. But here's the interesting part. Telomerase isn't active everywhere in the body. In most of our fully specialized or differentiated cells, telomerase activity is very limited. Its expression is highest in germline cells, the cells that give rise to eggs and sperm, as well as in certain progenitor cells, which act like reserves to replace old or damaged cells. Scientists believe this restricted pattern evolved as a defense mechanism. Nearly all human tumors show abnormally high telomerase activity, which allows cancer cells to divide endlessly. By keeping telomerase turned down in most cells, the body reduces the risk of uncontrolled growth, even if that means aging continues. But the balance is delicate. Too little telomerase activity can also be harmful. Germline mutations in telomerase-related genes that reduce its function are linked to serious stem cell failure diseases, such as dyskeratosis congenita, aplastic anemia, and idiopathic pulmonary fibrosis. These conditions reveal just how critical it is to maintain an optimal level of telomerase, high enough to support tissue repair and regeneration, but low enough to keep cancer risk under control. And it doesn't end there. Telomerase itself is regulated at multiple levels, from how much of the enzyme is produced, to when it's switched on or off, to how accessible it is to chromosomes. This tight regulation shows just how central telomerase is in the balance between health, aging, and disease. Of course, telomeres and telomerase aren't the only genetic players in aging. There are also the sirtuin genes, which act like a repair crew, helping cells manage stress, repair DNA, and control metabolism. In many studies, activating sirtuins has been linked to longer, healthier lives in animals. Then we have the FOXO genes, which are like guardians of longevity. They help protect cells from damage and regulate important survival pathways. Interestingly, people who live past 100 years old often carry versions of FOXO genes that seem to give their cells extra protection. And in nature, there are fascinating exceptions. The naked mole rat, for example, can live for over 30 years, an extremely long lifespan for such a small rodent. Scientists believe part of this comes from unique genetic traits that make them unusually resistant to cancer and other age-related diseases. All of this tells us one thing: aging isn't controlled by a single gene, but by a network of genetic programs. Some promote repair and longevity, while others limit growth to protect us from cancer. Together, they form the blueprint that shapes how, and how fast, we age. But genes themselves aren't the whole story. It's not only about the DNA sequence we inherit, but also about how that DNA is organized and regulated throughout life. This is where the epigenome comes in. The epigenome acts like a set of switches and markers that control which genes are turned on or off. It determines how tightly DNA is packed into chromatin and, in turn, which genes are accessible to be expressed. Studies in humans and in animal models of aging show that as we grow older, the epigenome gradually loses its original configuration. Chromatin, which normally has a well-organized structure, becomes more disordered. External factors, like diet, toxins, stress, and even lifestyle, can further disrupt this structure. These changes have profound consequences. They alter genomic integrity, disrupt chromosomal architecture, and shift patterns of gene expression. Genes that once protected the cell may be silenced, while others that promote inflammation or dysfunction may become more active. This epigenetic drift doesn't rewrite the DNA code itself, but it changes how that code is read, influencing both the rate of aging and the lifespan of the organism. In many ways, epigenetic change is like adding noise to the biological symphony of our genes. The music is the same, but over time, the instruments fall out of sync, leading to dysfunction across the body. But aging isn't just about these genes, it's also about the damage that builds up in our DNA. Every day, our cells are bombarded by radiation from the sun, toxins from the environment, and even errors during cell division. Over time, this damage adds up. When cells are too damaged, they sometimes enter a state called cellular senescence. In this state, the cell stops dividing but doesn't die. Instead, it sits there, releasing harmful chemicals that damage nearby healthy cells. In many ways, aging is the story of how our DNA slowly accumulates mistakes and how our bodies struggle to keep up. While genetics clearly shape some aspects of aging, large-scale genetic studies show that the effect of the genome on lifespan is modest. If our genes were the only factor, lifespans would have stayed relatively constant across history. But in reality, global human lifespan has nearly doubled in the last 200 years, while our genome has remained essentially unchanged. This points to something powerful: the enormous influence of non-genetic environmental factors. Epidemiological research has made great progress in identifying these exposures. Scientists have found at least 25 independent environmental and behavioral factors associated with premature mortality, biological aging, and age-related diseases. Among the biggest drivers of early aging and death are smoking, socioeconomic status and deprivation, ethnicity, levels of physical activity, and whether a person lives with a partner or not. Sleep quality, mental health, and physical well-being, including chronic fatigue, also play a central role. Even early life exposures matter. Things like body size and height during childhood, or whether a mother smoked during pregnancy, can shape health decades later. This evidence makes one thing clear: genes load the gun, but environment pulls the trigger. Our DNA may set the stage for aging, but the lifestyle and conditions we live in decide how quickly or severely it unfolds. So, can we do anything about it? Scientists are beginning to find clues. One fascinating discovery is that caloric restriction, eating fewer calories without malnutrition, can activate the sirtuin genes and extend lifespan in animals. It seems that when our bodies sense lean times, they switch into repair mode, protecting our cells. Researchers are also experimenting with genetic engineering. For example, using telomerase, an enzyme that rebuilds telomeres, scientists have been able to extend the life of cells in the lab. Some experiments in mice even show delayed aging when their telomeres are protected. But genes aren't the only factor. Epigenetics shows that diet, exercise, stress, and sleep can all turn certain genes on or off. This means that while we can't change the genes we were born with, we can influence how they behave throughout our lives. The future of aging research is exciting. Scientists are developing senolytic drugs that can destroy senescent cells, potentially slowing age-related decline. Others are studying stem cell therapies that might regenerate damaged tissues. And gene editing tools like CRISPR raise the possibility of directly repairing or enhancing our DNA. But this brings up a big question. If humans could extend life far beyond natural limits, should we? Would longer lives bring new opportunities or new problems? In the end, aging is written into our DNA. Our genes play a huge role in why we grow old, but how we live, our choices, our habits, and our lifestyles can shape how we experience that journey. One day science may unlock the key to slowing or even reversing aging. But until then, the best tools we have are already in our hands: caring for our health, protecting our mental, and physical well-being, and shaping the environment our genes respond to. If you enjoyed this video, don't forget to subscribe, like, and share to support more content like this. And let me know in the comments, if you could extend your life by 50 more years, would you want to?